Okay, so we'll invite the first group from Cochisin, all right, your Johnny, Chuiqing, and Xinjie to present on Nernst equations and equilibria. All right. Okay, so we'll welcome them.
Hi everyone. So today I'll be presenting on a bit of physical chem. So I'll be covering some concepts about electrochemical equilibria, and especially on the Nernst equation, which is something that I'm sure all of you have heard of before in H2, but maybe I'll never really use it. So what we'll be covering today is we'll first be covering the Nernst equation, what it is, as well as how we derive it. Then after that, we'll look at a bit more of the equation and see how we can apply it to different concepts related to equilibrium. And lastly, we'll look at some interesting electrochemical stuff, which are some useful electrochemical diagrams, which are the Latimer diagrams, cross diagram, as well as the Pobre diagram.
To start off, what is the Nernst equation? And there you have it. It is the Nernst equation in its full glory. Okay, so this is Nernst equation, and this is Nernst. He's staring at you, wondering, do you know how to derive it? And here we will derive it for you. To derive the Nernst equation, we'll be using the Gibbs free energy. The upper one is the equation we encounter in H2, which is of no use today. And the bottom one is the superior version of the Gibbs free energy equation, which we will use today. So let's begin with our derivation. First, Gibbs free energy is actually equals to the standard Gibbs free energy plus the thermodynamic constant times the thermodynamic temperature times the line of the quotient of reaction. And this quotient of reaction we'll touch on later. I claim that for electrochemical reaction, the Gibbs free energy of that reaction is actually equal to the minus of moles of electrons transferred times the times the cell potential and the Faraday's constant. The Faraday's constant is the charge carried by 1 mole of electrons. And by adequate algebraic manipulation, we can get the Nernst equation at the bottom. You simply divide both sides by nu s, and you shall get the according equation. The equation in the box needs further explanation. So what do I mean? How do we actually get there? So we'll get to the derivation of the derivation. We're considering an isobaric and isothermal reversible process. Now for this kind of process, Gibbs free energy is given by delta H minus T delta S. I hope you know what those symbols mean. We can further expand this equation by expanding delta H. Delta H is actually equals to delta U, the change in internal energy, plus P delta V, where delta V is actually a change in volume, plus V delta P, and delta P is a change in pressure, minus T ds. So what does an isobaric process mean? It means that the pressure doesn't change. So what does the pressure doesn't change mean? It means delta P is actually 0. So we get a new equation where the delta P term disappears. And the bottom we get delta U plus P delta V minus T delta S. We can expand the change in internal energy into heat, Q, expansion work, WEXT, and additional work, WADD. For an isobaric reversible process, the expansion work is actually the negative of PdV. So the two terms cancel out. And for an isothermal reaction, Q is TdS. So another two terms cancel out. All we have left now is delta G on the left side and additional work on the other side. And you might ask, what is the additional work? So we remember this is an electrochemical reaction. So I claim that all of the additional work is the work done by the electricity. And what is the work done by electricity? Well, it's Q times V, the charge times potential. Now potential is the cell potential. And Q is given by minus moles of electrons times the charge per mole of electrons. So there you have it. This is the derivation of the derivation of the derivation. And we have completely derived Nernst equation. Now Nernst is very happy. Okay, so next we'll be talking about the reaction quotient which Jim mentioned just now. So if you notice in the Nernst equation, you can see the reaction quotient Q. So if you recap what we learned in H2, the reaction quotient Q is described by this equation, which is essentially the product of the concentration of the products over the reactants raised to some random power depending on the equation that you see there. So what this means is that we can use this to predict what will happen in the reaction based on the Nernst equation. So let's say we introduce more reactants to this. We introduce more reactants to this reaction mixture. So my Q would decrease. So by the Nernst equation, you can see that my E cell will become more positive and the forward reaction, phase reduction, will be favored. So conversely, when more products are introduced, Q would increase. So E cell becomes less positive and oxidation is favored state. It's more favored relatively. Yes. So this proves this is the same as what is described by Le Chatelier's principle. So Le Chatelier is very happy. Yes. So in a special case where Q equals to K, so this is when the system is in equilibrium. The E cell will be equals to 0. So this means that we can use the Nernst equation to calculate the equilibrium constant at a given temperature, because we know that equilibrium constant varies with temperature. So you need to know the temperature before you can calculate this equilibrium constant. So as described just now, at equilibrium, you will get this version of Nernst equation. So you can substitute a Q with K, and you'll get this equation basically if you do some shifting of terms. So now we have some practice time. So take out your, oh my god. Oh really? Okay. So this is the question for you all to practice. We will give you all 4 minutes. So hopefully you all remember what Xinjie mentioned just now. It will help you in solving this question. Yes.
Yeah, okay.
This one.
Yeah, alright, so we want to show you the Nernst equation as well. So maybe I'll just want to copy down this question first. Very short question. Good point. You should include the Nernst equation.
You copy on Nernst equation, then
Good point. You just write on the equation.
Yes, right? You can write.
Oh my god.
You can write. Oh wait, I don't caption.
I think cannot go far.
Oh. I think we need more time.
Okay, I assume that everyone copied down this stuff already. Don't worry, just flash the equation so you can copy down the equation as well.
Yeah.
Okay. So also just a bit of a hint maybe. So this equation, the one in the red box, is also very useful to help you do your question. So I think this equation you actually should know. Yeah, but yeah, so just take note that this equation will help you. Yeah, so now we just move back to question.
It won't make noise here, but you're sitting through the other side. Oh, later. Yeah, later.
Oh, you want to use the speaker? That's. Can you all hear me?
Oh, yeah.
Can you all hear me? Okay, question 1, 2, 3. Calculate the standard half-cell potential of the following half reaction. Given that
So can use of course we will second version. So
Is it very, very long?
It's not like this. Also very important
Let me try to.
I'll finish my own interview. One more minute. Okay, so maybe I'll just give you all some hints as well. So like probably the first step you want to do is to actually find what is the standard potential of this reaction over here. Then after that, you can apply the Nernst equation to help you find what is the potential. Does that make sense? You know what? The question answer. Whatever, that makes sense. Never mind, never mind. That doesn't make sense. Like, sorry, you find the half cell potential of this reaction first. Then you can find the standard half cell from the Nernst equation. Yes. So because you self first, right? Basically, you have to go this side.
Okay, hopefully you all have gotten headway in the question. So I'll just start to go through, and maybe hopefully you all can do on the way in case you can. Okay, so right, this is the start of solution. So you can see that there are three equations here that we will use for the solution. So we ask you to find the standard half-cell potential of the equation. Yes, for this equation. Okay, so you have the first two equations to help you. Then after that, we gave you the we gave you the standard reduction potential of the first equation already in the question. So you can use that to find the reduction potential of the last equation. So because E cell is not an additive function as yes, but gives free energy is an additive function. So you can, it's a state function, so you can add them together. So this is by Hess law. So you can take the standard you can take the okay, you can take it gives you energy, or the first two equations add together, you get the standard gives you energy of last equation. So to recap, this is the equations that we are referring to.
Okay, yeah. So basically, once you get free energy of the equation that you're actually looking for, you also now you want to find what is the standard reduction potential of the last equation. So what you do is you convert. Basically, you want to use the values that are given to you. Basically, what the values you have are E1, you have E1, and you also have KSP. So if you convert, you can use basically like the equations and the delta G equals to minus NF E cell for this. And then you can also use delta G equals to minus RT along KSP for this to get the delta G in terms of the KSP. So from here, you can actually put everything together, and then you realize that, yeah, you divide everything by NF, which is in this case, 2 electrons are being transferred. So divide everything by 2F, and then you get this. So actually just from this equation, you can see it actually looks like a Nernst equation because it is. So from here, you just need to solve in the values, and you should get the answer. Yeah, so I hope at least you all understand. Maybe if you all don't understand like how to do the question, but at least you understand like what the solution is. Okay. So for those who managed to get this answer, congratulations, you are very good. Okay, so now we'll just be moving on and we'll just continue to talk about some more interesting stuff about electrochemistry. So look at some of these diagrams, which are very useful electrochemical tools to help us in reactions and doing problems. And they're the Latimer diagrams, Frost-Epworth diagrams, and Poblet diagrams. So I'll go through what each of these are. So this is the Latimer diagram right over here. So it looks a bit complicated, there's a lot of arrows everywhere, but actually it's one of the most simple diagrams out there. Because basically what a Latimer diagram is, it's just a summary of all the standard reduction potentials of an element. So in this case, this is a Latimer diagram for chromium. It goes from the highest oxidation state of chromium to the lowest oxidation state. So you have from chromium 6 to chromium 0. So all these arrows that you see over here are simply the standard reduction potential for this reaction. In this case, for example, chromium 6, chromium 5, this is just a standard reduction potential. Yeah, so this is just literally a summary of all the transitions. So it's very simple. So how you construct it is just you take out your data booklet and then later you can just form the values, you just put the values in and you can draw the diagram. So maybe like something interesting you can notice from the Latimer diagram. So let's say that you have the reduction potential from chromium 6 to chromium 5 and chromium 5 chromium 4, but you don't have from chromium 6 to chromium 4 the reduction potential here. So how do you find that? So you notice from the values over here that the E cell values are not additive. You don't just add up these two and then you get the reduction potential here. So why is this so? Is that if you notice that in all the equations related to E cell, the number of electrons being transferred in the equation actually matters. In this case, chromium 6, chromium 5, only 1 electron is being transferred. But then from chromium 6 to chromium 4, 2 electrons are being transferred. So the E cell value is actually related to the potential per mole of electrons. So adding this up presents a lot of problems because the number of electrons being converted each equation is different. So since it's such a hassle to work with E values, we can use something a bit more easier instead, which is our Gibbs free energy, because Gibbs free energy we know is a state function and can very easily manipulate it using Hess's law and just add them up. So to get these values, like in this case 0.95, you basically just need to convert all of this to Gibbs free energy, and then which will equal to the Gibbs free energy of the reduction from 6 to 4 and convert it back to E cell, and you get it. So some other stuff about Latimer diagrams is that Latimer diagrams will differ in acidic or alkaline solutions. If you see in this example with the Latimer diagram of sulfur, it's different in acidic and basic. And this is because in acidic and basic conditions, the compounds will exist as different dominant species. Yeah, so if in your data booklet, you also notice that the initial values will be different for compounds, whether an acidic or alkaline solution. So this is the same thing basically. So the latimer diagram will be different for acidic and alkaline. So what can you do with this latimer diagram? Well, first, it's a very neat summary of all the reduction potential for an element, if you're interested in. And also, it's very useful to help you construct force diagrams, which is the next type of diagrams. And this is a force absorption diagram, or simply just a force diagram. So if you notice, just on look, it looks a bit more complicated, but yeah. So yeah, I think was it 2018 or something? Yeah, something like that. So for the force diagram, US also is only one element. In this case, you see chromium. We have a force diagram for just chromium. And also, reduction potentials are different for the acid and the alkaline solutions also. So what is this force diagram? So basically what it does is it illustrates the relative stability of the different oxidation states of an element. So how is this shown? It's shown that compounds lower on the y-axis will be more stable. So why is this so? Well, I'll explain it. But to first understand why this is so, we must first understand what this diagram even is about. So we can first start by looking at what are the axes in the force diagram. So the y-axis we see is the number of electrons times the electrode potential, whereas the x-axis is the oxidation state of the compound. So in this case, it's from 0 to 6. So what is basically important about the force diagram is that the gradient between 2 points represents the standard reduction potential of the reduction reaction. So maybe for example, if we look at chromium 4 and chromium 3. So this gradient over here is a positive gradient. So what it is that this gradient is the standard reduction potential of chromium 4 to chromium 3. Yeah, so this is basically what this means. And it also makes sense actually, because if you look at the axis is number of electrons times electrode potential. Well, the y-axis is basically the number of electrons being transferred between oxidation states. So you take a gradient, which is this divided by y-axis divided by the x-axis. Basically, you also get the reduction potential. So yeah, it makes sense. And also to construct the diagram, you need to start from somewhere. And so what it starts from is that the element at the standard state will be taken to be 0 on the y-axis. So in this case, chromium metal is 0. And from there on, you can just plot the graph based on reduction potentials. So since the gradient represents reduction potential, it means the shape of the graph actually means something. So for downward slope, let's say in this side, downward slopes represent a negative reduction potential because the gradient is negative. So that means the reduction potential will be non-spontaneous because you know that negative E0 when compared to SHE means that I prefer to be oxidized instead, not going to be reduced. Yeah, so basically what it means that let's say for chromium and chromium 2, since it's a negative gradient, it means that the E value, the standard reduction potential value for chromium 2 to chromium is negative, so it's non-spontaneous. So therefore, basically the system will prefer to be this way towards chromium 2 instead. Whereas with the upward slope is basically the opposite. So a positive slope means that it's a positive reduction potential, means that the reduction reaction is spontaneous. So chromium 5 to chromium 4 is spontaneous. That means once in the species will prefer going this direction. So the end result is that since everything, basically the species just follows the shape of the curve, it will tend to move towards the lowest point. So this is why you say the lowest point is basically the most stable compound. And whereas if you're higher on the graph and more to the right means your oxidation state is higher, it also means that it is a stronger oxidizing agent because it has a greater tendency to reduce. Yep, so now that you learn all these force diagrams, it seems quite interesting, but what can you actually use it for? Well, actually one of the very useful tools of the force diagram is that it can be used to predict disproportionation just from the shape of the curve. So how do you do that? So let's take a look at the force diagram for hydrogen peroxide. So we know that hydrogen peroxide tends to disproportionate into water and oxygen. But how can you tell that from the graph? Well, if you just look at this graph and look at the gradient between these 3 points, we notice something. So you notice that the gradient for the reduction of H2O2 to H2O is, the magnitude is greater than from O2 to H2O2. So what this means is that, let's say we consider the reduction reaction from hydrogen peroxide to water, So standard reduction potential is actually greater than the reduction potential of O2 to H2O2. So if let's say we have a solution just hydrogen peroxide, and it can be either like, it can be both reduced or oxidized, then since the reduction potential of the reduction potential is greater than the reduction potential of the oxidation reaction, we know that E cell equals to E reduction minus E oxidation, then E cell will be positive in this case. So that means this reaction will occur and the disproportionation will occur. So just from looking at this convex shape of this graph, we can tell that H2O2 will actually disproportionate into water and oxygen. So this is one of the useful parts about the Frost diagram. So now we just move on to the last diagram, which looks the most complicated of all of them, and it also has the most complicated name. So it's called the Pourbaix diagram. Yeah, and it's discovered by some Belgian dude, but it's French. Okay, so basically what this diagram is, I think just by looking at the axis, you can sort of guess like what this is all about, which is basically it shows the possible equilibrium states of an aqueous electrochemical system as the pH varies. So what it does is that it maps out the reduction potential of different reactions involving an element. So for instance, this graph is some iron-containing compounds. It maps out how the reduction potential changes with pH. So actually, if you think about this, like how reduction potential changes with the concentration of some of the constituent ions, you actually be reminded of the Nernst equation, which is like how reduction potential changes with concentration in equilibrium. So basically, like what this means is that the Pourbaix diagram is really just a representation of the Nernst equation graphically. Okay, so some stuff to know about the Pourbaix diagram is that all the constituent ions and gases in the reaction, in this case like iron, all these iron-containing compounds are in their standard conditions. So for aqueous solutions, 1 mole per dm cube, then for gases 1 bar, except for H plus, of course, because you are varying the pH over here. So although the Pourbaix diagram is quite complicated, we're only really interested in the boundary lines between the different compounds. So there are really 3 types of lines, which are vertical lines, horizontal lines, as well as these kind of slope lines. So only really interested in the lines in the Pourbaix diagram. So I'll explain what each of these lines actually mean. So first, vertical lines. Vertical lines shows acid-base reactions. Like in essence, like you can see, like let's say for iron 2 and iron 2 hydroxide, like these reactions, like basically the dominant form in the solution will change depending on the pH. So this one is just a simple acid-base reaction. When your pH is higher, you prefer to be in iron 2 hydroxide. pH is lower, you prefer to be in iron 2. So this reaction, there's no redox involved. It only matters on the pH. So that's why it's just a vertical line. And the dominant form will just depend on the pH of the solution. So for horizontal lines, you realize that they have some reduction potential reaction, and that's because they are redox reactions essentially, but they're independent of pH. So for example, iron 3 to iron 2, this is a redox reaction that doesn't involve hydrogen at all. So of course, we expect that if concentration of hydrogen changes, the reduction potential should change. Yeah, so that's why it's a horizontal line. And the y-coordinate of this horizontal line is simply the reduction potential of the reaction on the graph. So the last type of line is a bit more complicated one, which is the slope lines. So slope lines essentially show redox reactions where hydrogen is a constituent ion. So hydrogen is involved in reaction. So for instance, with the reduction of iron 3 hydroxide, iron 2, so in the equation you can see there is a redox reaction, involves reduction, and it also involves hydrogen inside the equation. So what this means is that the reduction potential will actually change when the concentration of hydrogen changes. So how does it change? Well, we can look at the Nernst equation that we learned just now. So we look at the Nernst equation of this reaction. You can see that it's just Nernst equation, only 1 electron is being transferred, so it's just over F. And for the reaction quotient, since iron 3 oxide is a solid, water is a liquid, then we just ignore them. So we just get this as a Nernst equation. So we know that iron 2 is, we set it as 1 mole per dm cube, or pH varies. So basically, if you see this equation right over here, this is basically the equation of the slope line. Because the slope line shows the reduction potential as pH changes. Yeah, so literally the slope lines is just the Nernst equation put graphically. Yeah, so actually that's about all for our presentation. So I hope that you all learn a bit more stuff about electrochem as well as the Nernst equation. And maybe some of these concepts will be quite useful to you when you do a H2 as well, because you'll see a Nernst equation or any of these diagrams there often. Thank you.
Okay. Okay, thank you for the first group. All these slides will be available on Moodle. Okay, so the groups who have presented, please upload the slide on the Moodle. All right. Because of time, we will move on to the next presentation. Thank you so much. This electrochem Nernst equation is actually quite a useful extension. All right. One of the reasons why we have this student-led presentation, right, is so that because in H3 exam, sometimes they might ask something that is like a bit of more chemical principles, okay? So we want you all to be exposed to more of these so that in your mind, you get the overall picture of how certain topics can be extended, all right? So that's one of the purpose that we have it here. In fact, the materials as presented here, you sometimes see them in H2 Chem prelims exam. Yeah, so it's actually quite useful to know a bit more about this.
Okay, so a very good afternoon to all teachers, friends, and fellow schoolmates. So I am Pingray and Johan is beside me, and today we'll be introducing to you guys the principles of enzyme catalysis and biomimicry. Okay, I'll begin by introducing enzyme catalysis. First of all, what are enzymes? In H2 Chem, we have learned that enzymes are essentially just proteins and are biological catalysts. And proteins consist of a long chain of amino acids, each with its own specific R group. Enzymes may also contain non-protein components, such as coenzymes and cofactors, which usually contain metal ions. Now, the key feature of all enzymes is that all enzymes contain an active site, which is the region of the enzyme to which the substrate binds, and also the region of the enzyme which contains catalytic activity. Now, because all enzymes and all proteins have different amino acid sequences with different R groups, all enzymes have different active sites, and this results in the remarkable diversity of the number of different enzymes in our body. Now that we're done revising the basics, let's move to the fun part. How exactly do enzymes catalyze reactions? So in H2 Chem, we've learned the definition of a catalyst, which basically mean they lower activation energy barrier of a reaction. But how exactly do enzymes lower the activation energy barrier of a reaction? Let us explore with a simple illustration. Consider the breaking of a metal stick, which has to undergo a transition state to reach the final product. Now, if we sketch the energy profile diagram for this reaction, you'll notice that the activation energy barrier for this reaction is obviously going to be quite large as the transition state is unstable. So under normal circumstances, this reaction will not occur. Now let us imagine an enzyme with an active site lined with magnets. And this active site is specifically designed to bind to the transition state of the reaction to form stabilizing interactions with the transition state. So in this case, the stabilizing interactions would be the magnetic attractions between the transition state and the magnets in the enzyme. Now let us sketch the energy profile diagram of this reaction. It will look something like this. And if we put the two diagrams side by side, you will notice that there's a remarkable difference in the energy levels of the transition states of the two reactions. And this is because the enzyme stabilizes the transition state by binding to it, forming interactions and releasing energy termed as binding energy. And because of this stabilization, the catalyzed reaction will have a lower activation energy barrier, and hence the rate of reaction is sped up for the catalyzed reaction. The key takeaway from this whole illustration is that enzymes catalyze reaction by stabilizing the transition state of reaction and not the substrate itself. There are some questions down there for if we have time later.
Okay, now I'm going to introduce biomimicry. So biomimicry is how humans basically take structures of nature and to solve various complex human problems. So here are some examples. For example, there's this dried burdock fruits which have hooks and are able to hook onto animal fur or human clothing. And this is the inspiration for Velcro. And another example would be the big shape of the kingfisher. It makes it aerodynamic. And this is the inspiration for the nose shape of the 500 series Shinkansen. So our presentation, we hope to analyze how chemists take enzyme moieties, like parts of an enzyme, to synthesize catalysts for chemical synthesis.
I will now move on to the different modes of enzyme catalysis. In general, enzymes can catalyze reactions via a wide variety of methods, but for today, we'll just focus on the most common ones in our body, acid-base and metal-ion. Let me begin by introducing acid-base catalysis. As we have learned from organic chem, the presence of acids and bases can help to speed up the rate of reaction greatly by acting as catalysts. But in the body, we have a buffer system in our blood, which maintains the pH at around 7. And as a result, we have a low concentration of H plus and OH minus. To solve this problem, the R groups of specific amino acid residues in an active site can be acidic or basic, and can help to generate the H plus and OH minus ions needed to catalyze a reaction. Let us investigate a specific enzyme to deepen our understanding of this concept. Consider chymotrypsin, an enzyme that catalyzes the hydrolysis of an amide bond. Think of the enzyme as Pac-Man, while the pellets represent the substrates. Now let us zoom into the active site of this enzyme. Here we can see the three amino acid residues that are mainly involved in the catalysis of the reaction, aspartate, histidine, and serine. When a substrate is present, the nitrogen atom of the histidine residue is highly basic and will deprotonate the serine, the alcohol group on the serine residue. And the alkoxide formed is highly nucleophilic and will attack the amide bond. The role of the aspartate residue here is to increase the basicity of histidine. You all can think about why later on. Now, base catalysis also occurs in the next step of the reaction, where the nitrogen atom of histidine residue again acts as a base, this time deprotonating a water molecule. And the hydroxide ion formed is again highly nucleophilic and will attack the ester intermediate formed on the serine residue, and which will yield the final products. Because this trio of amino acid residues are so reactive and commonly found in many enzyme catalysts, they are collectively referred to as the catalytic triad. Now, recall that enzymes catalyze reactions by stabilizing the transition state of a reaction. In the absence of a base or acid, there's both a developing positive and negative charge on the transition state of reaction, and this tends to be less stable. Now, in the presence of an acid or base, one of these charges are neutralized. So, in the presence of a base, the positive charge is neutralized, while with an acid, the negative charge is neutralized. And this leads to a stabilization of the TS and hence a lower EA overall for the reaction, speeding up the reaction. Now, the cool part about enzymes is that the surrounding amino acid residues near the active site can also help to stabilize the transition state. So if we consider the base catalyst transition state on its own, here we can see the different possible interactions you can form with the surrounding amino acid residues, from weaker dispersion forces to stronger ionic and hydrogen bonding. And the beauty lies within the specific folding of the enzyme to maximize these stabilizing interactions. So for instance, if your substrate or if your molecule binding to enzyme is a large non-polar molecule, it is likely that the amino acid residues lining the active site of the enzyme will also contain bulky and non-polar R groups in order to maximize these favorable dispersion forces to stabilize the transition state.
So now let us see how scientists know this from nature. Okay, so some scientists, they synthesize this structure, So this structure, it contains the nucleophile, the hydroxyl from serine, the activator, the carboxylate from aspartate, and the base, the imidazole from histidine. And they integrated this into this huge macromolecule. Many copies of this in this huge macromolecule, which will speed up the rate of catalysis by this specific active site. Yes. So to analyze this, the scientists tested this enzyme to catalyze this reaction, the hydrolysis of 4-nitrophenyl butanoate to butanoic acid and 4-nitrophenol. And as you can see from this graph, the enzyme-inspired catalyst is quite effective as compared to the uncatalyzed reaction, showing that biomimetic structures do actually work. Thank you.
Moving on, let us discuss metal-ion catalysis, the next way of catalysis. So some enzymes do not rely on purely amino acids or do not consist of purely amino acids. And in fact, some enzymes are bound to metal ions. And these metal ions can catalyze reactions in a few ways. But for today, we'll just discuss the simpler ones. Firstly, metal ions can increase the acidity of a reacting species. This should not come as a surprise to many of you, as we have learned in H2 Chem that cations with high polarizing power are able to withdraw electron density from bonds in the surrounding ligands. And this can potentially promote the dissociation of H+, which can then undergo acid catalysis as previously discussed. Secondly, metal ions can also help to stabilize the charge of intermediates. Consider this enolate. In the transition state leading to this intermediate, there is a developing double negative charge, which is highly unstable. In the presence of a metal ion catalyst, such as copper 2+, the double negative charge is stabilized by the electrostatic attractions formed between the two opposite charges. And hence the TS is stabilized and there will be a lower EA overall for the reaction. And finally, the metal ions can also act as a center for reduction and oxidation reactions. And again, this should not come as a surprise to most of you, as we have learned about the roles of metal ions as homogeneous catalysts in H2. But in enzymes, the process of catalysis tends to be more complicated and sophisticated than just a simple one-step reaction we learn in H2. So consider the enzyme cytochrome C oxidase. For those who take bio, it's an enzyme found in the mitochondria. So this enzyme catalyzes the seemingly simple reduction of oxygen to water molecules. But do not be fooled by this apparent simplicity, because the full reaction looks something like this. Yes, so as you can see, the reaction is very complex and long. And let us just scrutinize the reaction by zooming into the roles of the metal ions here. We can see that the two metal ions here, iron and copper, are involved in basically every step of the reaction. And the roles of the metal ions can obviously be summarized. We can see that the ions act as redox centers, being oxidized and reduced repeatedly. And they also act to stabilize the intermediates formed in the reaction, which include superoxides, peroxides, and hydroxides. And finally, you can see there's also some form of acid catalysis going on here, with the H+ being converted to other H+. Yes.
Yes, let's go.
Okay, so some researchers are crazy enough to try to quantify this reaction by calculating the rate constant of each and every single step of the reaction, which is when they have actually succeeded. But the key takeaway from this whole illustration of this specific enzyme here is that enzymatic reactions are incredibly complex and complicated. And even till today, there's still a lot that we do not yet know about these reactions.
Okay, let's look at another metal ion catalyst, metal ion enzyme, which is iron hydrogenase. So iron hydrogenase is an enzyme that functions the same way as sodium borohydride or lithium aluminum hydride. Basically, it's a hydride ion donor. So this enzyme is found in organisms called methanogens. So methanogens, they derive the energy by breaking down carbon dioxide into methane. So this enzyme is particularly useful for us as it converts carbon dioxide, a greenhouse gas, into natural gas, fossil fuel. So before we actually dive into how this enzyme works, let's look at this concept called frustrated Lewis pairs. So in normal Lewis acid-base chemistry, the Lewis acid, electron acceptor, accepts an electron pair from a Lewis base, an electron donor, to form a bond. However, in frustrated Lewis acid-base chemistry, what we have is the Lewis acid, in this case, the pentafluorophenylborane, like this thing over here, yeah, has three bulky pentafluorophenyl groups. And the Lewis base, the triphenylphosphine, has three large phenyl groups. And these large groups, they sterically hinders a bond formation. Therefore, this bond, it cannot form. So yeah, so therefore the Lewis acid-base pairs are considered as frustrated. So what is the use of this Lewis acid-base pair? Is that essentially it helps to catalyze a lot of important reactions. Basically, it creates an electric field between the electron-rich site and the electron-deficient site, such that when small molecules enter between the field, it will rip it apart into atoms. Like for example, for hydrogen, you'll split it into a hydride anion and an acidic proton, allowing both of these substrates to exist in the same medium. Apart from the electric field explanation, some scientists have proposed that this reaction goes by a synergistic transfer of electrons. So let us go back to iron hydrogenase. So iron hydrogenase has a Lewis basic site, which is this O-, and the Lewis acidic site, this Fe2+. So this Fe2+ is very special. as we learned from H2, this Fe2 plus has a low spin state, which opens up two d orbitals for interaction with the O minus. So basically, this reaction center, it helps to split a hydrogen molecule into a H plus, which is now attached to the O minus to form a hydroxyl group, and a H minus, which is now attached to the ion center. And ta-da! This ion center now is a hydride donor. So what does it donate this hydride to? It donates it to the coenzyme as found shown on the left, which is methanol H4-MBT plus to form this reduced coenzyme, methanol H4-MBTH. And now this reduced coenzyme now acts like a hydride donor to reduce carbon dioxide to methane. And now let's look at the biomimicry of this enzyme. So a few scientists, they developed this new catalyst, which has a similar structure as iron hydrogenase. But instead of a OH as a basic group, now it has an amine group. And instead of a low spin D6 iron 2 center, it now has a low spin D6 manganese center. And to show the efficacy, they reacted it with this substrate on the left and found that the yield was quite good at about 92%. So what can we gather from all of this? Is that the communication between chemistry and biology is a two-way street. Chemistry helps shape our understanding of biology, in this case enzymes, and biology helps us develop better chemical methods to increase yield and efficiency.
I will now present a brief summary of enzyme catalysis. So firstly, enzymes catalyze a reaction by stabilizing the transition state of the reaction, and not the substrate, which is important. And secondly, enzymes can catalyze reactions via a variety of methods, which include acid-base, nucleophilic, and metal ion catalysis. In fact, most enzymes use a combination of the above methods in order to achieve its final goal of an efficient catalysis of all reactions. And finally, just a reminder that today's presentation is just scraping the tip of the iceberg of enzymatic chemistry, because there's still a lot we do not know about enzymes and science in general. And today's presentation serves just to hopefully give you all a better appreciation of biochemistry. Yes, thank you all for listening.
So this is in the previous syllabus, okay, because the previous syllabus is on pharmaceutical chemistry, all right? So there's a quite interesting application of chemistry, all right, in terms of medicine, okay? So I will not go through everything, all right, because we don't have time. So depending on time, I might just finish some examples on enzymes. So what are drugs, okay? So I just Googled this term, drugs, all right? And then this is the definition that came out to me, so I just copy and pasted it. So it's a medicine or substance which has physiological effect when you ingest it or otherwise introduce into the body, all right? Okay, is it on presenter mode? Okay, I suspect yes, it is. Let me just change it. Okay, all right. Okay, so now, then there are a few things that is interesting here. First of all right, it says that, okay, let me use a pen. Okay, so it says, first of all, you need to have, oops, okay, let me just get it. Okay, so first of all right, it must have psychological effect. Okay, not psychological, physiological effect. Means the drug, okay, can maybe cause you to feel better, cause your body temperature to drop, okay, cause your rashes to go away, all right? So these are the physiological effects. When ingested, so the medicine can be ingested or otherwise introduced into the body. So it need not be ingested, but it could be injected, for example, all right? So actually there's a difference. How come certain drugs, certain medicine you can eat, but how come certain medicine you need to straight away inject into in your body, all right? Because of the different properties of the drug. And then you see, I purposely choose this picture because one part of the medicinal chemistry is to identify how the drug can be ingested, all right, and how effective it is. So that's why you have different kinds of packaging. Certain drugs, this kind of most common one, solid, okay, crystalline form. Certain drugs cannot be crystalline, it has to be in the liquid form. Certain drugs, right, even though it is powder, it is a powder form, it cannot be packed into a capsule, into a solid pill. Then you have to have a capsule that encapsulates it. All right. So why is this so? All right. And why do you need that? Why can't you just eat the powder, for example? All right. So these are the questions, right, that scientists will try to investigate, all right, in medicinal chemistry. So when you eat medicine, where does the medicine go? How does the medicine work? Okay, does it work magically, like suddenly you eat Panadol, then you become well? All right, there's a process of the medicine. Medicine is just like your food. The route that the food go through, medicine will also go through. All right, okay, your food will eventually becomes your feces. Okay, your drug will also have the same process. If you are having higher dosage or higher amount of drugs that the body wants, okay, then you will just be excreted. All right. So one of the major targets of a drug, we call a target, means that the drug target where, okay, goes where, is on the cell. All right, definitely on the cell and parts of the cell. All right. So that's why cell is important. Okay. For bacteria, all right, the target is usually the cell wall. Okay. So what happens is that you eat antibiotic. All right, your doctor will prescribe antibiotic. What does the antibiotic do? The antibiotic is to kill the bacteria. All right, and the target of the antibiotic is the cell wall of the bacteria. All right, okay, but of course, not just bacteria. You eat Panadol. Panadol does not target the bacteria. All right, it targets something else in your body. All right, so therefore, the cell is where the drug targets, and the cell is not an open system. The cell is bounded by membranes. All right, bio students will be very familiar with these kind of pictures. And the membrane is very important because the membrane, right, is like two layers of surfactant come together. So outside of the cell and inside of the cell, it is water, basically aqueous system. But within the cell membrane, it is oil. So your cell has a layer of oil that protects it from the outside environment. So your cell membrane is basically like a layer of oil, basically, all right, that separates inside and outside. Because both inside and outside are aqueous. If you mix them together, they'll just be mixed aqueous, all right? So you need to have separation. And that separation is achieved by the phospholipid bilayer, and you have many proteins on the membrane. And these proteins could be enzymes, could be what we call receptors, and these are the ones that will that the drug, okay, will target. All right. Let you see how flexible this is. Okay.
Between the living machinery of the inner cell and the harsh conditions of the outside world stands the cell's plasma membrane. As crucial as this barrier is, it's surprisingly flexible. Push it and watch it move. Poke hard enough and it might break and begin to regroup. The lipid molecules of the membrane naturally assemble in a double layer because their tails repel water as their heads attract it. Throw in some cholesterol and a few carbohydrates, and you have the basic structure of a plasma membrane. Within these lipid molecules, we also find different proteins which do various things for the cell. For instance, they receive signals from the world outside, or they transport nutrients and waste. So nature composes the membrane with a combination or mosaic of different lipids, carbohydrates, and proteins. And these molecules are not stationary. They constantly move within the structure, fluidly changing their positions. The survival of all life rests on this veil of material. A supple membrane, just 2 molecules thick.
Okay, yeah. So all our cells in your body is just these 2 layers thick. So what are the 2 layers? Just like I said, right? So the tail is just your alkyl, non-polar alkyl chains. The head is actually a polar head with the nitrogen, all right? and the phosphorus. Okay, so this is polar. So the polar side is soluble in water, forming hydrogen bonds. All right. Then the tail is just non-polar dispersion force. So therefore, the tail is not soluble in water. So it will orientate itself to be with each other, the tail and the tail, whereas the head and the will just face the aqueous system. All right. So within the membrane, you have proteins, and these are the control system. So it controls, your membrane controls what goes in and out in your cell. All right. So this is called selectively permeable membrane. All right. So in order for the one to go into your cell, it must be someone that's for you. All right. So since my selectively permeable membrane led you through, you are the one. All right. Okay. Never mind. Okay. Next. Okay. So where does the drug interact? Okay. Basically, where does the drug interact on the cell? So the drug can interact on the proteins within the cell membrane. So it can just bind to the protein or the cell membrane. So that protein can be enzymes, just now introduced, or it can be receptor. All right. So that's the most common target for a drug. All right. So the drug will just interact. It goes to the active site or the non-active site of the enzyme and binds to it. It can also interact, goes into the cell and interact with the nucleus, interacts with the DNA of the cell. So one example of such drug is called the cisplatin. All right, it's an anti-cancer drug. It's one very famous example that was used in your. I'm not sure. I think still in your notes. Okay. So we'll look at a video on cisplatin. Okay, later. All right. So this cisplatin, right, it targets the DNA of your cell. So this medicine, all right, if you take it, it will go to your nucleus, and then it will affect how your cell can reproduce, because your DNA is the one that controls all the protein production. All enzymes is produced by the codes in the DNA. So if you destroy the DNA, you destroy the process. Okay, so let me share this video. This is just a very funny video that I found previously online, all right, that on mechanism of the cisplatin. So I'll just share this video. So it's a cell membrane, okay? So you see a lot of proteins are floating on the cell membrane. Then you see an army of cisplatin drugs coming in. One of the organelles in your cell. Yeah, this is actually what happens in your cell. All right. It might look very imaginative, okay, but it is what it is. Okay, yes. Of course, this is a dramatized version of what is actually happening, all right? So, but what is true, okay? What is true is that this drug, all right, targets the cisplatin, targets the DNA, all right? So what happened is that it will bind to the DNA, okay? And then it will cause the enzyme to be unable to attach itself to the DNA, and therefore you cannot have the processes that the cell wants to do, all right? So that is the way cisplatin works. And what else the drug can target? The drug can target the cell membrane itself, all right? So for example, you have this drug, okay? It's a fungicide. Fungicide means it kills fungi, all right? It's called amphotericin. What happens is that you have this molecule that has both non-polar and the polar group. So the non-polar group is able to form dispersion force with the non-polar alkyl group of the phospholipid. And then you have the polar group which is able to form the hydrogen bond with the ecosystem. So what happens is that it can attaches itself into the cell membrane and it force open a channel. So what happens is your cell is now leaking through the channel that's being formed by this drug. And if that is the case, right, what happens is that inside your cell, you have very high concentration of solute. So water goes into the cell, your cell will become swollen, all right, just like me. And then eventually, okay, it will burst, okay, which I hope I won't happen, okay? So when the cell bursts, you have the cell is dead, all right? So therefore, you have effectively killed, okay, whatever infection that's caused by the fungi, for example, all right? So that's how, so different drugs will target different parts of the cell, all right? Okay, so that would be the three different targets of the drugs in the cell, all right? This is just a summary. Okay, I'm not sure if I want to continue. Okay, maybe I'll just share the most important thing. Now, drugs has to be very, now, organic chemistry becomes very important in designing drugs because your enantiomers are different in terms of the spatial orientation. So R isomer and S isomer will interact differently in an enzyme, for example, because your active site of an enzyme is very, very specific. So your different isomers, even R and S, as simple as R and S, just the swap of the bond will cause it to be unable to fit. So one example, all right, is given here. It's just here is a drug. It's called noradrenaline. All right. So this noradrenaline, if you look at the diagram, let me just see. Okay, no. All right. So if you look at this diagram, right? Okay, this molecule is designed very, very well. All right, to fit the active site or the groove nicely. How does it fit nicely? You have the OH group, the phenol, right? It forms hydrogen bonding with the probably serine, all right? The OH group of amino acid on the enzyme. Okay, then you have this part, right? NH3 plus can form very nice ionic attraction with the side chain on another amino acid that just nice position it such that you can bind the molecule properly. And then if you look at this chiral center, the H is pointing out, the OH is pointing in. And just nice, the OH that's pointing in, right, will form a hydrogen bonding with the top OH group, okay, on the enzyme. Now imagine instead of this isomer, you have the other isomer. If you have the other isomer, right, means that the H is inside, the OH is outside. So if you have the H being inside and the OH being outside, the OH will be too far away to form hydrogen bond with the OH group on the enzyme itself. So therefore, what happens is that you don't have optimal intermolecular interaction, all right, intermolecular forces, and therefore this drug will not bind it well. It will just leave. So this drug will not have any effect, okay, on this enzyme, for example, all right. It will just go in, it doesn't fit, so it will just come out again, all right. But if it fits well, okay, it will stay there, and then subsequently you will have maybe reactions, or you will just block this place. So your drug, okay, I'm not going through the enzyme because your friend has gone through just now. So your drug can act as blocking. So finish in 5 minutes. Okay. So your drug is reversible inhibitor. For example, what it means is that your drug can mimic the natural substrate of an enzyme, and then you just occupy the enzyme. So if the drug occupies the enzyme, it will block the actual thing that's supposed to bond with the enzyme, and therefore it will cause the reaction to not happen. So for example, for this particular bacteria, all right, so you have particular enzyme that actually uses this PABA, all right, for aminobenzoic acid as a substrate to bind with the enzyme for a reaction. Okay, but then what happens is that if you consume this particular drug, okay, so the sulfonamide, if you consume this drug, right, if you see the molecule looks very similar, NH2, NH2, benzene ring. The difference is that this is CO2H, this is SO2, NHCH3, but both of them are polar. So it means that you can, and both of them contain C double bond O and S double bond O, very similar. So you can cheat the enzyme thinking that I'm actually binding to the correct substrate. So once the binding happens, okay, it blocks the enzyme from the actual correct substrate that the reaction has to occur. So then you can stop the enzyme from functioning. So that is why how your antibacterial could work. So sulfonamide is one kind of bacteriostatic, means that it can stop the bacteria from growing. The bacteria didn't die. Okay, so it's not a penicillin. The bacteria didn't die. The bacteria just cannot replicate. All right, and then it can be just excreted, and then it will just die naturally. All right, so you didn't kill the bacteria at once. You just stop the bacteria from multiplying, okay, from replicating. So that is one way your medicine works, okay, by killing the, how does it work with the bacteria. Now, another kind, which is more permanent, just now it was less permanent, sarin, nerve agent. I think you all heard about this before. So what happened is that this sarin, right, once it's a drug, okay, of course, but it's not of good use, okay. Once you ingest it in your body, what happens is that the sarin will form a permanent covalent bond with the enzyme, all right. So normally what happens is that you have acetylcholine, all right. Normally this enzyme is able to catalyze the hydrolysis of the acetylcholine, all right. So then you will catalyze the hydrolysis of the ester. But once you have sarin is able to fool the enzyme, all right, and you will form a permanent covalent bond with the enzyme. So what happens is that enzyme will be blocked. It cannot perform its function to catalyze the molecule. So acetylcholine, why is it important? Because acetylcholine causes muscle contraction. So if you want to do this, okay, if you want to stand up, walk, okay, read or do anything, you need muscle contraction, right? So you need acetylcholine. Now, this enzyme is breaking down the acetylcholine when you don't need muscle contraction. But by blocking this enzyme, your acetylcholine cannot be hydrolyzed. So you forever have a lot of acetylcholine. So what happened to you? You'll forever have muscle contraction. Okay? You'll forever have muscle contraction. So what happened is, spasm basically. And you will contract until you die. Okay? That is why is it called a nerve agent? Why is it called sarin? So who was killed? Kim Jong-nam many years back, all right? 3 years ago, all right? He was killed by something of a similar nerve agent, not sarin, all right, but it's a DX nerve agent. So this nerve agent also bonds permanently with the enzyme. So he basically died of muscle contraction, all right? Died of continuous muscle contraction because of so it works quite similarly, all right, to the sarin. So you see, drugs can do good things, drugs can do bad things, all right? So, and depending on what you take, all right, it has a effect. I think I'll stop here. This is only like first part of, I mean, if we have chance next time, I'll introduce a bit more, right? But here is just a very brief introduction, okay, of a